Good afternoon. You're all very welcome to this special session on unlocking the potential of industrial yeast for production of high-value consumer products as part of EFIB 2020. So, hopefully my first slide is appearing. Great. So, my name is John Morrissey. I'm based at University College Cork in Ireland, and I'm the session moderator today. And we've got a panel of speakers who you see listed on the right there, and I will introduce these in a moment. So this particular session is it's organized as a satellite session at EFIB based on outputs from a European Union funded project. So I just want to acknowledge this project at the beginning. It's called CHASSY, and I want to acknowledge the funding of the European Union for this project. And it's funded under the pillar of leadership in enabling and industrial technologies. And it's based on microbial chassis platforms. And there are other projects also funded under this particular topic. The focus of our project was on the development of efficient cell factories for industrial production of novel high-value products. And it's a yeast-based project. And we have a consortium of academic and industrial partners that you can see listed in the center of the slide there. And these are led by different scientists, some of whom will be very familiar to people working in the yeast biotechnology field. Now, yeast has been used for a long time in biotechnology. On the left of your slide, you can see some traditional products that come from yeast biotechnology. Fermented beverages have been produced for thousands of years. And some of these other products have been produced for well over 100 years. In the center, you can see some of the more recent products based on recombinant DNA technology, which have been produced from the 70s, 1980s, up to now. And you can see a lot of these are biopharma based. And more recently, there's been a lot of development of products for the bioeconomy. But predominantly the only products I would say that have really reached the market are bioethanol, lactic acid, succinic acid, produced by some big biotech companies. Flavors are something that's also emerging onto the market. But one thing that's interesting when you look at this progression is what you can see is back at the traditional products, there's actually a wide range of different yeasts have been used for their production. This is only a subset of the yeasts that you see there. Whereas once we came to the recombinant DNA era and the modern era, it's largely Saccharomyces cerevisiae has been the workhorse of the biopharma and the biotech industry. And of course, there's very good reasons for that, not least because of the availability of recombinant DNA technology for Saccharomyces cerevisiae. But one of the things that we have been very interested in is to see whether, as well as Saccharomyces cerevisiae, some of these other yeasts, which maybe were involved in traditional production or not previously, might have potential in the bioeconomy as industrial hosts. So Saccharomyces cerevisiae, but also other yeasts for the bioeconomy. Now, why would we be thinking about that? And one of the reasons is that we all know that there's huge potential for yeast cell factories. You possibly can't read the detail here, but the message is that there have been many different prototype or proof of principle yeast cell factories published in scientific literature. This is a review article from from Verena about 5 years ago, and that listed a lot of different molecules and different classes. But despite this, very few have ever made it to the market. And the question is, why is that? And it's because unlike maybe the products like bioethanol or even things like lactic acid, which are very simple pathways, the products that we're interested in for modern biotechnology are highly complex and they require a lot of metabolic engineering. So strain construction is very complicated. It takes a long time to develop a strain and it's really expensive. And these are the challenges that we've been trying to overcome in the CHASSY project. And we've had some very good success. And in the course of this session that I'm introducing here, you're going to hear about some of those success stories. And in terms of an overall outcome, what we've developed are innovative platforms and toolboxes that will accelerate the capacity to bring new products to the market in certain classes of molecule. And we're going to hear about those. And this will involve the use of Saccharomyces cerevisiae, but also other yeasts. So over the course of the next 5 talks, which are all going to be quite short, we're going to hear first from Jasmijn Hassing and then from Florian David on the product classes where we've made some big advances, and these are aromatics and oleochemicals or lipids. Then you're going to hear from myself and Tristan Rossignol on the work that we've done on developing new platforms, new industrial yeast platforms with Cluvomyces and Eurovia. And we will hear from Verena Siewers towards the end of the session, where she will talk about some of the new approaches and how we can really bring a systems biology perspective to strain engineering. Before I pass over to Jasmijn for her talk, just to give a little bit of housekeeping, the idea is that we will get this session, we will have all the talks delivered by about 16:45 or so Central European time, and that gives us 15 minutes for discussion then. So if you have a question for any of the speakers, or indeed a comment as we go through the session, please type it into the chat function. The moderator, which is me, and excuse me, pardon, the moderator and myself will collate these questions. We'll ask them to the particular people in the panel session afterwards. So we won't take questions immediately after each talk, but please type your question in as we go along. So I'd like to just, I'm not going to introduce every speaker. I'll let them introduce themselves, other than say first speaker here now is Jasmijn Hassing from Technical University of Delft. Thank you.
Oh, sorry. Okay, great. Thank you, John, for introducing me. Yeah, so like you said, my name is Jasmijn, and I'm a PhD student at the Technical University of Delft, and I'm also working on the CHASSY product. And I will tell a little bit more about the plans and the results that we have obtained so far concerning the production of aromatic compounds. Or actually how I normally phrase my PhD project as one ring to make them all. And of course, we're not thinking about like an elfen ring that's meant to dominate the world, but we're more thinking about an aromatic ring, because this aromatic ring is actually present in a lot of different products. And you can really find it back in compounds such as fragrances or in flavor compounds, but also therapeutic medicines or even commodity products such as rubber. They all contain this aromatic basic structure. And this also means that there's actually a huge market for these different products. So here you see a few examples. I mean, we all know vanillin, but there's also a lot of food applications, so as food fragrances or food flavors or colorants. So phenylethyl acetate, for example, quite a product that's used a lot, but not super expensive. But you also have flavonoids, which is kind of a growing market because you have thousands of flavonoids. And back in the days, they were mostly used as food colorants. But now there's also a growing interest to use these compounds as therapeutic medicine because they show a lot of benefits as they show antioxidant behavior. They are possibly anti-carcinogenic. So there's also like a growing interest in producing these compounds, but also more common fragrances such as phenylethanol, but also products as lignans that are used as food preservatives. And what you can see is there's really a huge market. So the market ranges from a few tons to $1 million US. And also the selling price goes from relatively cheap for phenylethyl acetate, but also some of the compounds can be up to $100 per kilogram. So there's really a big field and there's a big interest for these products. But the problem is that most of these compounds are nowadays produced via plant extraction. And this has some downsides because, of course, you're quite limited if plant extraction is your main source because you're dependent on a good harvest, but you're also kind of limited in area. So you cannot produce as much as maybe the market demands. What's also quite difficult is that plants normally don't produce just one aromatic compound, but they use several. And it can be quite hard to just isolate one. And quite often you really need a pure compound for your application. And extraction is also quite difficult. So this is also why quite a lot of these compounds are very expensive because the yields are very low. But what is the main advantage of plant extraction is that these compounds are considered natural. And I think it's been said a few times already during EFIB that the consumers nowadays they really want natural products. So they're also really willing to pay for that. And that's, of course, a huge downside of chemical synthesis. Because, I mean, chemical synthesis has more potential to produce these compounds unlimited and stably. So especially when you look at therapeutic medicine, that's really important to always have stable supply. But a lot of these aromatic compounds are quite complicated, as John already mentioned. So you quite often have a lot of different subgroups. You have a lot of decoration molecules. It's really hard to produce these compounds chemically. And not all these production methods are very sustainable. And of course, these products are not natural. So when you want to use these products in food, you can already not really sell them anymore. So this is why we really thought there is a problem and there's kind of like a need to fill this void to produce these wanted chemicals, but in a better way. So that's why we're looking at microbial production, because this should potentially offer a stable and unlimited supply of these products. It has more potential to produce pure compounds because in principle, if you have a very specific enzyme, you can only make one product. Of course, this is not always the case in theory or well in practice, but there is more room for diversity for very specific enzymes. Products produced via microbial fermentation are also considered natural and can be sold as such. And so what's important to know is that actually a lot of these aromatic compounds that I talked about, such as flavonoids, they're all derived from two main substrate sources. So we have, of course, we need the aromatic ring. So they're always derived from aromatic amino acids, so mostly phenylalanine. And quite often they require melanin coenzyme A. So these two compounds, these serve as the main substrate for any of these other aromatic products that we're interested in. So actually what the main goal is of CHASSY within this field is actually to make a yeast chassis strain. So that's also why the name's CHASSY, that is really efficient at converting simple sugars into this precursor, so that we have a strain that has a lot of this precursor, which can serve as a precursor supply for any of these products that you're interested in. So it's basically kind of like a plug and play. You just take whatever product pathway that you want and you put it in our yeast chassis, and then you can really focus on how your product pathway is behaving. And you don't have to worry anymore about the precursor supply. So you can really just see, is my product pathway working and where are the limits there? Instead of still have to, engineer to put 2 years of strain investment in just the precursor before going to the product that you're really interested in. So for that purpose, we've constructed 3 different yeast chassis. So John already mentioned them a little bit. Of course, we're working with Saccharomyces cerevisiae because it's easy to work with. The tools are there. And we've managed to construct a strain that produces over 1.5 grams per liter of the precursor. So these are titers that are really already getting industrially relevant. But indeed, we're also focusing on a bit more exotic yeast. such as Cryphomyces maxianus. And this strain is mostly interesting because it's thermotolerant, so it can grow up until 52 degrees. So this means that it's very relevant in an industrial context because you don't have to spend so much money on cooling anymore because this strain can survive at higher temperatures. And we're also looking into Yeasts lipolitica, which is mostly known because it can store really high contents of lipids. And also for these two yeast chassis, we have succeeded in reaching like 0.5 gram to close to 1 gram of liter of the precursors. And this is all in a batch characterization. So if you would go to, for example, fed batch, you can even increase these titers more. So this is really, they're really behaving good enough to really start looking at product and expressing genes to get these products. So there's not a lot of science in this presentation. It was more of showing you what we have and what we can offer. So if any of you have a product pathway that you would like to test, but you don't have a suitable platform strain, then we have several chassis yeast strains that you can use. Of course, we also have a lot of know-how on the strain engineering. We've spent, a lot of people have spent a lot of time on getting these precursor supply to these levels. So there's no need to reinvent the wheel. So we also have this knowledge to share. And of course, we're also very open to collaborations. So if you would like to hear more or discuss collaborations, then I will also be available in the breakout room, or you can send me an email. Thanks.
Jasmijn, thank you very much for that nice overview. I see some questions are coming into the chat, so I'll hang on to those for later. So I'm just going to move straight on now to our next speaker, who's Florian David. And Florian is going to talk about the second major product class that we've been working on in the CHASSY project. So Florian, I think you're there. If you are, the floor is yours.
Thank you. So the presentation is loading. Great. So thanks for the intro. So my name is Florian David. I'm the CSO of Biopetrolia. We are a startup company focused on microbial production of fatty acids and derived products as well. And we're also part of the CHASSY project. So the focus on this presentation is on yeast platform strains producing fatty acids and also fatty acid derived products we have developed within this project. So first of all, a little bit of around the motivation. So why do we look at microbial production of fatty acids? And actually we have identified 3 major reasons to do that. And the first one is that there's a very high demand of fatty acids, and they can be found in a lot of different products, from food, personal care, to many other products actually, also including biofuels, of course. But we have a problem because most of the fatty acids are unsustainably sourced, for instance, from palm oil, as we all know, which leads to deforestation and also majorly contributes to climate change and loss in natural diversity as well, of course. In addition, there are actually there's a wealth of fatty acids out there in nature which can simply not be sourced and easily extracted, because it just, let's say, you can only find them in very rare sources, let's say rare plant sources, for instance. And the idea is here also that we can use synthetic biology and microbial production to really kind of tap into this potential and scale the production of all these compounds out there, which actually have great opportunities to look into. So to tackle all these challenges I already mentioned a little bit, we are focusing on producing fatty acids with using yeast cell factories. And we use synthetic biology and metabolic engineering to really engineer the yeast cell factories and to convert very efficiently the feedstock, which can be, for instance, sugar-based, into our product of interest, serving all kinds of different markets. We identified 3 key advantages to use microbial fatty acid production. The first one, it's more sustainable, or it's actually sustainable in itself, based on a variety of feedstocks, including also waste streams and side streams, which can be used by our microbial hosts. and which then also, of course, leads to reduced land use and also reduce in greenhouse gas emission, where other technologies kind of really foster this. Secondly, we can really engineer our yeast cell factories to make tailor-made structures, including, for instance, the fatty acid chain length or saturation, and assemble also these kind of fatty acids into complex products with specialized properties and then also specific applications. The third point I want to bring up here is that microbial production is also a cost-efficient alternative production for fatty acids, especially specifically derived products. Of course, this is always dependent on the products you look into, but already today we have actually microbial production, ways of microbial production, which is already cost competitive in certain areas. So how do we do this? So to reach cost competitiveness, and here we need a lot of optimization, as John already pointed out. There's a lot of time needed and money to do that, but we're getting better and better in this actually, and also we're getting quicker in doing these kind of optimizations to get really efficient cell factories. This is here just a sketch of this. What we also see quite often, these typical design, build, test, learn cycles, where we really iteratively optimize our cell factories. And during the CHASSY project, we actually have developed a lot of new tools, but also ways of actually analyzing our cell factories to make them better and more efficient, so that we actually are able now to kind of get strains which produce in the gram per liter range. Also, to mention this here again, we have also different organisms providing different advantages. First of all, Baker's yeast Saccharomyces cerevisiae is a very industrial robust host, already proven to produce a lot of different products. It's well characterized and easy to engineer. Whereas on the other hand, we also have Yerovia lipolytica here, which exhibits natural high fatty acid production, which is also of benefit, of course. So back to the fatty acids as a product. So fatty acids are actually starter molecules for a lot of different products categories, ranging from fuels, nutraceuticals, to food ingredients, fragrances, but also cosmetic ingredients, and also agrochemicals, just as some examples here. And these are used across a wide spectrum of market segments for a wide array of applications, serving $1 billion industry. So there's a lot of potential out there to replace these fatty acids. And again, to mention this, a lot of these products suffer from lack of sustainability, poor environmental impact, and also very high costs in production. And fermentation-based processes can offer really good solutions to these kind of problems. And we can also kind of tap into the potential of novel products that are so far not easily to produce in a scalable way, or just have very high costs associated with them. So within the CHASSY project, we actually have developed platform strains producing high amount of fatty acid precursors and fatty acids of specific chain length. And just to summarize this here and maybe create some appetite for you, what we have developed here are platform strains producing C16, C18 fatty acids like palmitic and oleic acid. But in addition, we also really engineered these strains to move to the different end of the spectrums. So we actually have platform strains which produce very long chain fatty acids, so longer than C18. As an example, proof of concept product, we have here the Rossignol, which is a very long chain fatty alcohol used for various applications in cosmetics, but also as a pharmaceutical. And then on the other end of the spectrum, we have short chain fatty acids, so shorter than C16. Here we also have developed platform strains, and as a proof of concept product here also, we have actually strains producing octanoic acid and the derivative hydroxyoctanoic acid, used also in applications for food, personal care, and pharmaceuticals. So also to mention this here specifically again, we have developed these platform strains, and they're very available now and very good starting points to develop a variety of fatty acids and fatty acid derived products. And this allows us, for instance, to easily do a proof of concept for, let's say, a very long chain acid or fatty acid or short chain fatty acid derived product, and then we can quite quickly see what we get in our yeast cells, and then use our platform strains in a plug and play manner, so to speak. This brings me to my last slide. So please feel free to reach out to us if you're interested in this technology, potential collaboration projects, or also licensing opportunities. And contact Biopetrolia. So this would be Anastasia and myself. So here are our email addresses. And also for the short chains, fatty acids specifically, please feel free to contact Eckhard Bolus, who is the responsible for this part of the project. So thanks for the attention and looking forward to the Q&A later.
Thanks very much, Florian. That was, again, a very nice overview. $billion markets out there, so big money to be made for industry. And hopefully your company will get a little bit of it. So I'll introduce the next speaker now, which is myself. So I think my presentation is appearing here. I'll thank Jasmijn already for telling me that I work on an exotic yeast. I've always wanted to be exotic, so that's very nice. But what I'm going to talk about is the work that we've been doing in CHASSY to develop this yeast, Cluvermyces marxianus, for food and industrial biotechnology applications. And I suppose one thing just to mention for all of the presentations, pretty much all of the projects or the applications that we're talking about here are ones that are led by particular groups, but there's several groups across the consortium have been involved, and this is no different, even though I'm presenting on behalf of the team. Most people have heard of Cluvermyces yeasts, but maybe not Cluvermyces marxianus. People might be familiar with K. lactis, Cluvermyces lactis, but K. marxianus and K. lactis are commonly associated with dairy products, kefir grains, different kinds of milks and cheeses. And because of this, Cluvermyces has got EFSA, European Food Safety Authority status as qualified presumption of safety, and it's got GRAS status in the US. And this actually makes it quite attractive for applications in food and biotechnology. Now, Cluvermyces marxianus, in fact, is already commercially used for a couple of quite well-known applications. One of them is the production of bioethanol. It's not the major source of bioethanol, of course, but for well over 20 years, or maybe even 30 years, Cluvermyces marxianus has been used in what's known as the Carberry process for fermenting whey permeate to ethanol. And Cluvermyces marxianus is very good for this because it's a natural lactose utilizing yeast. And this is a commercial process that's still ongoing with the Carberry company. A second application that has been commercialized is from the INRAE company, which is a subsidiary of Lecafre. And this is essentially a bioconversion of phenylalanine to 2-phenylethanol. Jasmijn already talked about 2-phenylethanol. It's one of our kind of proof of concept aromatic molecules as well for de novo synthesis. But natural phenylethanol has got it's got a niche for quite high value market. There's obviously a lot of chemical phenylethanol produced as well. So K. marxianus is already used industrially. Now, some of the reasons, actually the reasons why it's used in those two processes are really because of its very efficient production of aromatic molecules in the second one. And in the first one, because it can assimilate lactose. And in fact, it can assimilate a wide range of sugar sources. It's very fast growing. And as Jasmijn mentioned, it's thermotolerant. So it's exotic. But there are challenges and limitations, and I want to mention these because that's why it hasn't been, of course, more widely used in industrial biotechnology. And these challenges include limitations of the knowledge of strain physiology, and ultimately that feeds into a lack of scale-up experience and expertise. But also there's been a lack of molecular tools for strain engineering. People haven't known what host strains to use, and there haven't been good examples of engineered strains to actually demonstrate the application. So in CHASSY, we set about trying to address some of these limitations with a view, of course, to developing K. marxianus as an efficient industrial biotechnology platform. I'm not going to talk about details of the data other than to say we spent a lot of work looking at strain physiology of different strains to kind of figure out how strains would perform in bioreactor type conditions. A lot of chemosat cultures and other bioreactors. And so we have a lot of knowledge now and know-how around the physiology of this yeast that we didn't have previously. And we did a lot of work on developing the the methodology and the technology for strain engineering. And that's molecular tools, and it's also deciding which particular host strains to work in. This is, of course, topical this week because we use CRISPR-based genome engineering. Very disappointed not to be included on that list of Nobel laureates this week, but anyway. Telling jokes when you're looking at a screen yourself isn't quite as effective. But anyway, that's a joke. But like everyone in industrial biotechnology, we were developing CRISPR-based genome engineering platforms. And I just put up a couple of things here just to illustrate that we've published some of these platforms, and we've made a lot of parts available in open access repositories. And the reason why we've made everything available like this, it's part of our sort of open innovation strategy, because a lot of what CHASSY is about is developing know-how and tools that can be exploited by a wide range of companies. Of course, within the project, we also have some proprietary products and processes that are linked to the industrial partners within CHASSY. But from the point of view of the academic partners, we're kind of going down the open innovation route. And so we are making our tools and resources available. But of course, what we've got, which is maybe a bit unique then, is we've got great know-how on how to actually apply these. And as an example, Jasmijn mentioned phenylethanol, and I mentioned it myself in my introduction. This is a product that comes from the metabolism of aromatic amino acids. It's probably not the most complicated pathway, but there are still 8 or 9 steps involved in the biosynthesis of this molecule from simple precursors. And we've used the platform that I mentioned to sequentially engineer strains, and this is what you're looking at in the right. This is just titers of 2-phenylethanol and then tyrosol coming from strains through a progressive engineering strategy. The details aren't so important, but this is an example of how you can engineer a strain that if in the very first bar there's nothing, it doesn't produce any phenylethanol, to producing 750 mg per liter in batch culture. So this is something that can be applied to other aromatic molecules as well, or indeed to other classes of molecules. So the reason why we think this is of interest to industrial partners is we've got a host with a wide substrate range. It's now amenable for genetic manipulation. It's got interesting physiology that I didn't talk about. And from CHASSY, we've got the know-how to construct these cell factories, a lot of knowledge now of genetics, physiology, and metabolism. And we're very interested in collaborative partnerships with industry to see how we can help you if you've got processes, in particular based around aromatics, but also maybe other classes of molecules in Chloromyces marxianus. So thank you very much. And I'm going to immediately pass over to the next speaker, who's going to be Tristan Rossignol from INRAE in France. And Tristan is going to talk about the other non-Saccharomyces platform that we're developing in the project, and that's Yearovia lipolytica. I'm talking here just while I see Tristan appearing. So Tristan is here. So Tristan, the floor is yours.
Okay, thank you, John. Good afternoon, everybody, and thank you to attend these CHASSY sessions. I'm working in INRAE in France, so the French National Research Institute for Agriculture, Food and Environment. And as a member of the CHASSY consortium, our lab was involved in developing Yearovia lipolytica as a platform for biolipid productions. So each yeast in this project has its own specificities for as a potential industrial platform, and I will describe a little bit why Yearovia is interesting. Sorry, here. So Yearovia lipolytica is known as an oleaginous yeast. It means that it can accumulate a high amount of lipids, and you can see on this slide, on the picture, that it can accumulate lipids on structure called lipid bodies with quite a high concentration. It can go up to more than 70% in some conditions. Another trait that is interesting, it can grow on hydrophobic substrate like alkane, oil, and fatty acid, and so on. And that's why it has been mainly isolated from lipid-rich environment like polluted soil or cheese also. Two other points that it's important to mention, as John said for Marcianus, Yarovia has the GRAS statute from the FDA and also the QPS statute from the European Food Safety Authority. So this yeast has acquired this statute not only because it has been used as for academic research, but also because it has been used for actually quite a long time for industrial purpose. And I just pick up a few examples of industrial application in different sectors. It's not an exhaustive list, but just to tell you that it has been used long ago. It has been used by British Petroleum, like in the 70s, to produce single-cell protein from fossil resources, thanks to its capacity to grow on hydrophobic substrate. Much more recently, it has been used as a feeding for animal farms by the Polish company Skotan, and well, more related to our subject here, DuPont companies use it to produce omega-3 using a highly modified Yarovia lipolitica strain. So this late example show that this yeast is a good choice for lipid production, but this is not only because it can accumulate a lot of lipids. It's also because this yeast is well known for its lipid metabolism. So in fact, it has been used for decades now as a model organism for lipid metabolism, and so its physiology in this particular aspect is very well known. So it's a good starting point to produce sustainable and eco-friendly biolipids. However, the genetic engineering to produce non-native product and biolipids, as mentioned John in the first presentation, it's very time-consuming, quite expensive, and there was really a need for optimizing strain and to develop a new generation of genetic tools to unlock the potential of this yeast. And this one was, of course, one of the objectives of the CHASSY consortium. And in this regard, we developed CHASSY strain, Yarovia lipolitica CHASSY strain, optimized for some of the precursors of the lipids, like acetyl-CoA or manolin-CoA, which are the building blocks for lipid synthesis, by modifying the metabolic pathway, the endogenous metabolic pathway. And we developed tools to plug new pathway, mainly heterolux pathway, to produce specific biolipids. And the idea is to improve the quantity of lipids and to control the composition of the lipids we want to produce. So same strategy has been used for aromatic also, as shown for Carmarcianus. So such development has only been possible by setting up new versatile and efficient tools for dedicated to synthetic biology. And there is, of course, CRISPR, for instance, that we have now a set of tools that allows genetic editing of CHASSY strain, of course, but also in any genetic background now, even wild-type strain. But more importantly, we have developed pathway assembly technology using Golden Gate to develop multi-gene pathway. So in this kind of technology, the idea is to assemble a large set of DNA fragments in one pot reactions. And we have now a large set of DNA fragments that is also available in AGENE as an open access. And that allows to build up multi-gene pathway in one reaction, and we have a large set of promoters now that is available. Today, thanks to this new technology and thanks to the former technology also, we have developed a CHASSY strain optimized for precursors for lipids, and we can plug pathway now very efficiently and very rapidly. First, to use different carbon source, like carbon source that Yarovia is usually not able to use naturally. And you have a list of possible carbon sources now on the left side of the slides. And of course, we can plug in pathway for production of very specific compounds. And here, for examples, biolipids. And I just show a short list of unusual lipids that we have be able to produce as proof of principles, like orchid fatty acids, racinoleic acids, cyclopropane fatty acid, and so on. But you can also produce other derivatives from the acetyl-CoA or malonyl-CoA, like carotenoid production. So you can see here on the picture, there's a big orange bioreactors here. So that brings me to my last slide. So I hope I convinced you that Yarovia is a good chassis for biolipid production, thanks to its potential for biolipid accumulations, for all the synthetic biology tools that has been developed, but also thanks to its GRAS statute and its capacity, its robustness for industrial conditions like low pH that I didn't mention before. And of course, we are glad to set up any partnership for strain development using this strain. And you have our email address on the back of the slides. So thank you for your attention, and I give back let the room for the next speaker.
Okay, Tristan, thank you very much. That's a very nice overview. Just to let everybody know, the talks are being recorded and the presentations will be available afterwards. So if you don't quite get the email addresses written down, they'll be available, or you can always find us on CHASSY, the CHASSY website, and indeed in various pages on the EFIB website as well. You can see more information about some of the outputs that are being discussed. So I'm going to pass over now to the final speaker of this session, who's going to be Verena Siewers, who's going to talk about some of the newer innovative ways that we are going about strain engineering. So Verena, you're welcome, and the floor is yours.
Thank you very much, John. So my name is Verena Siewers. I'm from Chalmers University of Technology in Gothenburg, Sweden. And I will talk to you today about how we use computational models to improve our cell factories and platform strains. You have already been introduced to the design, build, test, learn cycle, the iterative cycle that we apply in metabolic engineering. And in the design step of this cycle, genome scale metabolic models are actually a great asset because they can improve and accelerate the strain design to predict gene targets for strain modification to improve the production of your strain of interest. Just to give you a slight idea how such metabolic models, or the classical type of genome scale metabolic models, look like. So in these models, you have each gene and reaction represented in a stoichiometric matrix. So these models are purely stoichiometric models. And what can you do with such models? So you can take these genome scale models of your organism of choice and, for example, introduce a pathway leading to your high-value product of interest. And then you can feed the model, for example, with glucose or xylose as a carbon source. And the model allows you to determine the maximum biochemical yield, to evaluate alternative pathways, estimate internal flux distributions, but most importantly, of course, to predict beneficial genetic manipulations to further engineer your strain of interest. So we have in the past, for example, used such a genome scale metabolic model to select gene targets, in this case for CRISPR-based regulation. So our aim here was to increase malonyl-CoA production as a crucial precursor, but with a final aim to increase the production of 3-hydroxypropionic acid. So with help of this genome scale model, we predicted 168 gene targets for either upregulation and downregulation, and could show or prove that the manipulation of these gene targets indeed increased the production of 3-hydroxypropionic acid. However, these classical genome scale metabolic models have two major drawbacks. So the first drawback, or the first disadvantage, is that they don't account for spatial constraint. So there is, of course, a limited amount of total protein per cell, and with this also a limited amount of each protein or each enzyme. but the enzyme mass is something that is actually not accounted for in the classical type of metabolic model. The second constraint is that there can be variation in enzyme efficiency. So if you take this example of two alternative pathways that lead from glucose to your product of interest, one pathway, the one via enzyme Y and Z, requires less protein, but has a lower reaction rate, so would be slower, whereas the other pathway via enzyme A and B needs more protein mass, but has also a higher reaction rate. So what we did now within this project is to develop an approach called GECKO that includes enzyme amounts and catalytic enzyme efficiencies into this model. So the classical genome scale model becomes an enzyme constraint model, which then improves the predictability of these models. So we have developed these enzyme constraint models for our 3 chassis strains or yeasts: Saccharomyces cerevisiae, Cloiveromyces maxianus, and Yarovia lipolytica. But of course, this approach also allows us to develop enzyme constraint models for any other organisms for which we have a classical model available. So what these enzyme constraint models now allow you to do, they allow you to predict interventions, genetic interventions, such as deletion of non-essential enzymes, but also fine-tuning of levels of key enzymes. For example, if the model predicts that in order to achieve high product yields, you need high amounts of the green enzyme, whereas your experimental measurements tell you that there is only a small abundance of the green enzyme, the model will tell you to upregulate the expression of this enzyme, and vice versa. For example, if you have high abundance of the purple enzyme, but you only need small amounts to reach high product yields, it will tell you to downregulate this enzyme. So we have now developed also not only a pipeline to develop these models, but also to use these models to predict gene candidates for either overexpression and knockouts. So it's a pipeline that involves several steps, including optimization of flux. It takes enzyme usage into account, but then, of course, the mechanistic interventions such as knockouts and upregulation, but also it tells you which genetic targets are actually compatible with each other. And using this pipeline, we have now predicted gene targets for 110 industrially relevant chemicals, such as alcohols, alkaloids, aromatics, amines, fatty acids, flavonoids, and so on. So in case you, for example, are interested in further developing your own platform strain or your own yeast cell or its cell factory of choice, feel free to contact us, me or my colleagues, and we will be happy to initiate further collaborations with you. Thanks a lot.
Thanks very much, Verena, for showing us there the power of genome scale models. So we're going to move over now to the panel discussion. So I think now I'm probably waiting for panel members to appear in front of me. Yes, I've seen the panel members appearing. I'm getting an echo on my microphone. I'm not sure why that is. We've had a few questions come into the chat, and I'm going to pass those questions on to people now. If other people have questions, please type them into the chat. I can see in the chat as well there's breakout rooms after the session, and you can see the breakout rooms for the different people. So just after we finish the panel discussion, if you click on the relevant breakout room, if you do want to chat to one of those people privately. If for some reason you don't get into the breakout room, it might be packed out, tickets are hard to come by, in which case you will be able to contact the people privately anyway, through the channels I mentioned. Okay, so I'm going to pass on a couple of the questions that came now. So I see I've got Verena, Jasmijn, Tristan, and Florian all available. So one of the questions from Adeline talked about is related to the applications, and the question really is for the applications in the food market. What's the situation with using genetically modified organisms? And Yasmin, I'm going to be directing this question towards you, because I think in your talk you kind of refer to some of those applications, and in the context of natural molecules and so forth. So would you like to comment on that?
Yeah, of course. Yeah, it's a bit of an interesting regulation, because I've looked through the documents of the European Union, and they state that you can sell a product as natural as long as the substrate that you use can be found in nature. So in our case, it's glucose, so that's a natural substrate. And as long as your product can be found in nature, so for example, phenyl ethanol can be found in nature, so it's a natural product. So as long as the substrate and the product can be found in nature and you sell it completely separated from the organism, you can sell it as a natural product, at least up until this point. So in that sense, you can sell it as a natural product.
Okay, thanks, Yasmin. Does anybody else on the panel want to comment on that? Put up your hand if you do, and we will unmute you. Okay, so I don't think we have any more comments. I guess one comment I would make is that this whole designation of natural is quite complicated, and just like with GMOs, the regulations vary, around the world. So it is something I know that causes companies a certain amount of challenges, and development of products in this regard is also market related, I think. Okay, so I'm going to ask this question now. I'll ask the question first, but I'm going to be asking it to Florian and to Tristan in the first instance, and it relates to the use of sugars, because I think just looking at the question, yeah. So the question really is whether it's likely that strains engineered for these high-value products, I'm paraphrasing now, but these strains engineered for high-value products, will they be able to produce from non-glucose substrates?
Yeah, maybe I can kick off here. Yeah, it's a good question. I mean, there's a lot of research out there also to really move strains towards more complex and also waste stream related carbon sources. So the idea here is that you will be able to use these kind of waste streams to also produce your product of interest. But I would say, so in connection to high-value products, maybe you not necessarily actually need to do this shift, because you can actually afford kind of high production costs when you produce high-value products. But on the other hand, if you look for something which is more, let's say, low-priced and more in a bulk fashion, then you really need to go down with your production costs. And one way to do that is to really look at your kind of feedstock you use and really bring down the cost via this. I hope this answers the question. But Tristan, also feel free to comment as well.
Yeah, I agree with you. The cost of your final product is quite important. I mean, the higher it is, the less you need cheap carbon sources, but there's still some, like for Yarovia, you can use glycerol, which is quite widely available, and it works pretty well. Well, that's what I can add also. I see it's not always a limitation, as you say, that if your product is high value, the cost of your carbon sources is not so such a limitation.
Thanks, Tristan. I might just ask Verena to comment on this as well, because I noticed that in your model, in the image you showed, you had xylose and glucose. And can I just ask it kind of, I know it's a very general question, but in terms of looking at a modeling for production in the pathways, does one see dramatic differences, let's say, in the pathways or in the yields or potential yields, depending on what sugar one feeds in? I see Verena is still muted, so if we could unmute Verena, please. Thanks.
I would say that depends very much on the product pathway as well. So it has, and then also it depends on the path that you choose from the sugar towards your product. So because of course there's issues with carbon balancing, cofactor balancing, ATP balancing. So that's a lot of factors. In some cases it may play a role, in some cases it may not play a role.
Okay, thanks. I'll answer my next question myself, but then I'll see if any of the panel want to comment on it. And it's from Heiko, and he says that I said that the strains that we develop within CHASSY are open for industrial application, but what about the strains that we used? Are they also free for industrial use? I think he means that, you know, the actual strains that we showed you in the presentations here. And I guess what I'd say is that most of what we showed you here were based on published material that we published, often using resources that were already available in the scientific community. What we've really got of, and though lots of those are pretty much routine and available for anybody to use. In some cases, we have developed very specific strains, and those would be at the moment in-house for the partners who have them, maybe for particular applications, and they could be available for collaborations. And then as well, a lot of what's really valuable is the know-how to actually engineer specific pathways into strains, because very often people will have particular host strains that they want to use for applications. And so the know-how that we demonstrate on prototype strains can easily be ported into another platform. Would any of the other panel members just like to comment on that point? Or are you happy with, stick up your hand if you do. I don't see any hand coming up. There was another question earlier from Pedro. I'm just going to ask the question and then look for a volunteer to answer it. But the question is, given the fact that we mentioned several grass organisms, Cluvermyces, Eurovia, Saccharomyces, is there any prospect of using these and chassis platforms for developing therapeutics like living drugs? So I think the suggestion is, what about engineered strains that produce biomolecules for, I'll say, in vivo application, maybe for consumption? And I can see all my panels squirming, so I'm going to ask Jasmijn that question.
Do you mean combined with the microorganism, or if there's also potential to make therapeutic medicine in our platform strains?
I think the question is more combined with the organism. So, for example, could you have your Saccharomyces producing, I'll say, a flavonoid, and you get the person to eat the Saccharomyces? I think that's the question.
Well, that's a bit more difficult. I would say, of course, you know, when it comes to medicine, people are less opinionated about GMOs. So, you know, as long as it can save their lives, then GMOs are less of an issue. But of course, the legislation is very, very strict, especially in Europe. So I don't think the rules are there yet to really the GMO yeast while it produces the medicine. I think it's much easier to really just separate the molecules from the yeast, and I think that will be much faster to implement than getting the public to accept.
Yeah. Tristan, did you want to speak, or?
I can rapidly talk,
yeah.
At least for Eurovia. As far as I remember, the European statute for safety, it's only for the product of Eurovia and not Eurovia itself. So I think it can use for animal forms feed, for example, but for human, I think it's not yet accepted.
Yeah.
That is case of therapeutics protein productions from Eurovia, but not the microorganism itself.
Yeah, I think that's correct. I mean, for Kluvramyces and Saccharomyces, they are also approved for consumption. And indeed, there are probiotic strains of Saccharomyces and Kluvramyces on the market, but they're not engineered strains, they're wild-type strains. I mean, what the question kind of relates to probiotics in a way. And even with bacterial probiotics, which are probably a much more developed industry, I don't think that there's examples of engineered probiotic bacterial strains yet, lactobacilli and bifidobacterium, even though I know it's a very active area of research. I'm conscious of time, but we have another couple of questions, so I'm going to try and put them on there. This is a question for Verena from Edgard. And the question is whether the modeling tools that you've developed can also be used to improve complex traits like thermotolerance or inhibitor tolerance.
I would say that the model that I presented today, the type of model, that would not account for temperature tolerance. But we have activities or had activities within our group as well to develop different type of models that also, for example, take temperature-dependent properties of proteins into account, such as like protein stability and activity, temperature-dependent activity of enzymes. So that model could actually be used to predict temperature tolerance, for example, or was also shown to make predictions about which enzymes to, for example, overexpress to enhance temperature tolerance. Inhibitor tolerance might be a little bit more difficult. It could be like if you have a metabolic pathway to degrade the inhibitor, then it could be integrated here as well. If the mechanism of toxicity of the inhibitor is more complex, then it might be more difficult to predict it with, or make predictions with such a model.
Yeah, or I guess as well, if an inhibitor inhibited a certain pathway that you might also, a metabolic pathway, you might be able to think about overcoming that. Okay, so I've got a question here for all presenters from Kirsten. Let me just read it. Okay, so it's asking, it's a general question, so whether any of the strains suffer from decreases in productivity due to essentially inhibition by the product. And if the answer is yes, it asks about whether you use an overlay in the fermentation to remove this inhibiting effect. So I guess it's asking whether we're using, for example, in situ product removal or something like that. So I'm going to ask Florian, because he's been staying very quiet. But Florian, have you come across either an examples you showed us there or other cases where you have had product inhibition, and maybe how have you tackled that?
Yes, sure. So there are different ways of how to tackle this. So one is, of course, to have some kind of in situ product removal. What we also have seen in industry, sometimes even this kind of dodecane overlayers to take out the product. But then, of course, there are a lot of strategies as well around engineering the cell factory in itself. So, for instance, there are these approaches where you use adaptive laboratory evolution, where you expose your strain to the product you're producing, and then you do this kind of transfers and really kind of select for a strain which is more tolerant towards the product, which you then can use as a starting strain, for instance, or you really look into the details, what's going on with the strain, why it is more tolerant. Then there are also all kind of other, let's call them synthetic biology or metabolic engineering strategies you could use. For instance, you could change your final product, put something on it that it's not that toxic anymore, and you can actually revert this kind of modification afterwards. And then there are also approaches where you, for instance, overexpress transporters to export the product that it's less toxic. So there are a lot of strategies actually out there to combine this.
Okay, thanks, Florian. Tristan, do you want to make any comment about product toxicity in Eurovia and whether that's been an issue for you or how you've dealt with it? You're muted at the moment. Yeah.
Yeah, we faced that sometimes for like I don't mind which product it was, but we apply strategy to remove the compounds in situ, but we did that with partners. So I don't have the exact details how it was made exactly, but I know partners who did that with specific bioreactors where you can do that. And well, I don't know what I can add to Florian, because I said you can do you can evolve your strain also to, as it's been mentioned before. And well, I know sometimes with fatty acid or fatty alcohol, or you can hydroxylate your fatty acids to make it less toxic, for example.
Yeah.
Also.
Okay, thanks. First part was suitably cryptic, so I think Tristan knows how to do it, so maybe talk to him privately if you want to be recovering your fatty acids. Okay, because we've passed the 5 o'clock Central European time, and I don't see any more questions on the way in. I'd like to thank the speakers for the presentations and working in this format. I'd like to thank Nathalie in the background from the EFIB, who set this up and done tremendous work getting everything going. Thank all the participants. We had about 60 people in listening to the talks, so thanks for taking the time and for your questions. And I'll thank the Europa Bio, of course, for organising the conference. I saw that Agnes was in there earlier, and I thank the EU for funding our research, and hopefully they will continue to fund industrial biotechnology lavishly in Horizon Europe. So that brings this to an end. And as you saw, there are breakout rooms. The links are in the chat on your right. So I actually don't know how I get into my room, but if you click on my link, you'll end up there. Or if you want to talk to any of the other speakers. And as I say, if you don't manage to talk to us, please send an email. You'll find us on the EFIB site, on the CHASSY site. Thanks very much.